Hi, I'm Robyn Stacey Humphreys. I've been a radiologist for over 30 years, and actually I specialize in cancer imaging, both CT, CT, and actually mammography. So ironically, in 2011, I became the thing that no physician wants to become. I became an interesting case. So I was diagnosed with diffuse large B-cell lymphoma in my late 40s. I don't have any siblings, so I had no great match for allogenic stem cell transplant. So I actually researched other things, and I found out about CAR T-cell therapy. I searched myself. I found a trial that had an open space in several states away. I flew there, I was immediately accepted, and in September of 2016, I received CAR T-cell therapy, one of the few first 100 in the world for lymphoma. So that was in September 2016, and here I am.
My name is Stephanie Trong, and I actually, went through non-Hodgkin lymphoma treatment myself back in 2017. And so this is a topic that's very near and dear to me, and really excited for our panelists today. But really my own experience as a cancer patient is what motivated me to want to start something to really help humanize the cancer experience, treatment information, all of those things for those of us who don't have a science background, right? So that was the genesis of the patient's story. We reach cancer patients, their care partners, caregivers with content, mostly, in-depth stories. We also highlight conversations with specialists in the field, like the amazing people we have tonight who are featured. And the goal is to humanize cancer. What does that mean? It means you're not alone. And here's some information we hope will be helpful. And our partners tonight, the Leukemia and Lymphoma Society, or the LLS, which has done incredible work definitely across blood cancer space in terms of research and community. The focus again is the latest in DLBCL treatments and research. And so for our doctors tonight, we have two who are specialists in lymphoma, and one who is a 3-time DLBCL survivor. So first with Doctors Brody and Phillips. Dr. Brody is coming to us from Mount Sinai, and Dr. Phillips is coming to us from City of Hope. I know, Robyn, first name basis now, because I consider you a friend. This is a great segue because you started off, you spent years on the medical side, a radiologist, you're seeing patients, and then you became a patient yourself. So with that being said, let's set the stage a little bit here. First, DLBCL, we hear about it a lot. Just a quick summary, but Dr. Brody, could you just sort of talk about what is DLBCL and what is the standard care for the first line treatment?
Sure. Big question. I'll try to give a quick answer. DLBCL, diffuse large B-cell lymphoma, is the most common. When I say the most common, we say the highest incident lymphoma in America, in the world as well. And so in America, about 90,000 people every year diagnosed with lymphoma. About 25,000 of those are diffuse large B-cell lymphoma. This is so frustrating to patients because in some way you just say, lung cancer is lung cancer. That's not even true, but it's a bit simpler. Lymphoma, you guys have all heard this, super complicated. We have about 84 types of lymphoma. And then people say, wait, do I have the good one or not? It's not just 2 types. It's not the good one and the bad one. We divide these lymphomas into many ways so we don't have to memorize a list of 84 things. The first way we divide them is first into, as you know, Hodgkin's and non-Hodgkin's. We are talking about non-Hodgkin's lymphoma. We divide non-Hodgkin's lymphoma into B-cell and T-cell lymphomas. So we're talking about a B-cell lymphoma. And then we sometimes divide B-cell lymphomas into low grade versus aggressive. Low grade, these grow very slowly, follicular lymphoma, marginal zone lymphoma, some others. And aggressive lymphomas, diffuse large B-cell, again, the most common of them. Maybe you've heard of Burkitt's lymphoma, mantle cell lymphoma, some of these other things that can grow more quickly. And The treatments for lymphoma, thankfully, we're so lucky because they have evolved more rapidly for lymphoma than for any other cancer in the world. Even though we are the 5th most common cancer, lymphoma, we have more FDA-approved medicines than any other cancer, more than lung cancer, more than breast cancer. And the reason is because thankfully the progress has been very rapid. We don't get FDA-approved medicines without progress to prove that those therapies can be pretty good. The standard therapies for diffuse large B-cell lymphoma historically over the past 20 years has been, we always have these alphabet recipes, RCHOP. It's not great branding, the R-CHOP. It sounds a little aggressive, and it is, as Robyn can tell you, it is a bit aggressive. People that are working full-time before R-CHOP are frequently working part-time during R-CHOP. So it's aggressive enough that it affects your life in a real way. It affects your hair, as you saw from Robyn's pictures. And R-CHOP is aggressive, but not the most aggressive stuff we have. It is a standard therapy. Even today, it may not be the only standard frontline therapy. There are a couple of slightly new versions of R-CHOP that are possibly options now. And I will say that in 5 years from now, I don't think R-CHOP will be the standard. I think we'll even have some better things. But R-CHOP, I'm not going to go into all the details, but the R, you've heard of this thing called rituximab. It is an immunotherapy targeting lymphoma, but using your immune system to kill those lymphoma cells, but that are not powerful enough by itself. So the CHOP is just regular old chemotherapy. The P is prednisone, so you've heard of that and not too tough, but the rest of that CHO is chemotherapy. All the side effects you've heard about with chemotherapy, hair falling out, a little bit of nausea, not so bad, but it's a real thing. And some of these other side effects you've heard about. The big punchline of our CHOP is, thankfully, it cures about 60% of people with DLBCL, not every type of DLBCL, but overall DLBCL. That's pretty good. We would say that people with, metastatic breast cancer don't really have a cure. And so we would say we're lucky to be able to cure 60%. Not that lucky if you're in the other 40, though. As Robyn or lots of our patients could tell you, we are very lucky for that other 40. We actually have a lot of progress and probably still some curative things.
Well, I appreciate that, Dr. Brody, because first of all, you outlined it really well. I know there's a lot to cram in there in just a couple minutes. But I do want to ask Dr. Phillips to lay out what are the options and also what does it mean, right? Like if somebody especially relapses in the first, is it 6 months, 12 months? How different is that, prognosis or treatment options?
I will say for patients who do not respond to R-CHOP and immediately relapse, what we consider, primary refractory patients, and that likely extends out at least into the first 6 months. I think those patients have done very poorly historically with a lot of the standard options that we have, which are other alphabet soups like rice or DHAP, which are just various combination of chemotherapy drugs, hopefully that are not sort of cross-resistant to each other, provide a little bit different mechanism of action compared to the drugs used in CHOP, with rituximab as well, with the goal of getting into an autologous stem cell transplantation. Again, historically, that group has done fairly poorly because they are probably chemo resistant, meaning that they're not going to respond to any sort of chemotherapy drug, because the whole design of chemotherapy is to prevent DNA replication by causing DNA damage at some point during the replication cycle. So if a cancer drug has figured out a way to get past one, it's likely to figure out a way to get past most of the rest. So, and then an auto transplant should be named an autologous stem cell rescue because it's still just more chemo. So it's not anything fancy about the stem cells that you're getting back. They're just your own stem cells to rescue your body. I will say for the most part, for a lot of the clinical trials that tout these relapse refractory regimens, they're probably dependent heavily on these relapse patients, which are generally the patients who relapse a year or so after chemotherapy. These are the reasons why we get the response rates for most of these regimens, such as, the rice and the DHAP, the big Coral study, which was a big regimen looking at these relapse refractory regimens to see which one was better. I mean, they're really probably driven by refractory patients again, relapse patients, because these refractory patients are not the ones who are going to drive these studies. So a lot of big emphasis recently has been on other ways to treat these patients, especially these early refractory patients or patients who relapse within 6 months to a year with other regimens besides chemotherapy, which brings into the CAR T discussion, which I'm sure we'll get into later. For patients who can't get to CAR T or who are ineligible for CAR T, I mean, in the second line setting beyond chemotherapy, there is lenalidomide and tafasitamab, which are for patients who are ineligible for autologous stem cell transplantation. That regimen itself, at least from the clinical trial, seems to be more effective in those who have only receive one line of therapy versus those who are heavily pretreated, meaning they have 2 or more prior lines of therapy before receiving this drug. Really in that second line setting, I mean, there aren't really a ton of other agents approved, even though we have options such as polatuzumab, bendamustine, rituximab. I'll bet that regimen is a bit hesitant. We are a bit hesitant to use that regimen in some sense because the bendamustine part does cause quite a bit of depletion of your immune system, specifically T cells. And the T cell depletion can be quite profound and prolonged, which can hamper things that depend on T cells to be effective, a la CAR T, et cetera. But there also is a new agent. Recently, there's a CD19 drug antibody conjugate called loncastuximab, which has a very different drug attached to it. It's a pyridynamine drug attached to it, which is a little bit different from MMAE, which is our usual antibody drug target. So that is also another option. And then there's selinexor, which is exportin-1 inhibitor, which was approved in this patient population, but probably has very little uptake because of the toxicity of the agent. So, but if we're looking at currently approved agents, to see the chemo or the regimens I just mentioned are the currently only approved ones outside of CAR T at this point.
I just want to go to Robyn again, to humanize this experience, because you've been through a lot of what was already discussed, different kind of chemoimmunotherapy regimens. You also went through an auto stem cell transplant. And then you eventually went into CAR T, which we will get into. But how did you, so it sounds like you were the one who researched CAR T. I wanted to ask, how did you bring up the discussion of CAR T with your hematologist-oncologist? And what were the differences for you in terms of going through the stem cell transplant and then going through CAR T?
Overall, I did extremely well. And that said, this auto stem cell transplant was extremely difficult for me. I was very, very sick and terrible experience. I did survive. I'm here, but I didn't want to go through that again. And as far as CAR T is concerned, when the auto stem cell transplant, which is really, again, it's just high dose chemotherapy, it's really a rescue, failed, I did not want to go through an allo transplant. So I had gone online. There's a site called www.clinicaltrials.gov. Anybody can get online. I really had no advantage being a doctor, except that I had heard about the site. And so I had started looking at different trials. And my husband, who's an engineer, he and I both looked at all these different trials and emailed investigators when I relapsed. And so we just tried to find a space in a trial. I just thought that was the right decision for me. It was not recommended by my oncologist. As a matter of fact, my oncologist thought that I should go ahead and have an allo transplant using my son as a donor, as a haplo. And I didn't want to do that. Personal. So to answer your question, I looked at it on my own. I think nowadays things are very different. In 2016, there was only one phase 1 clinical trial for Kymriah, which is what I had. So I based my decision on 26 patients, and 12 went into remission, which is not a big number. My oncologists were not comfortable with that, but somehow I felt that was the right decision for me. Everyone is unique. I did get different opinions from other oncologists. It was a very complex case. I was a younger patient who was healthy. So it was a decision, but I was so sick during the stem cell transplant with a septic shock, and that helped determine what I decided to do. Everyone is unique and it's a big decision, but I wanted to live my life and not have the risk of something called graft versus host disease, which could actually limit the enjoyment of life. And so that was also within my decision. So I'll leave it at that.
No, and I think it's a great example of, again, the equation, right? It's people, and for everybody, it's a different equation in terms of what you weigh more heavily. Is it how long you think you can, get more out of life in terms of survival, or is it quality of life? And Doctors Brody and Phillips, just curious, hearing Robyn's story there. I know that this happened back in 2016, so it's a very different landscape than now, but anything you want to add in terms of the decision-making or talking about this with a patient?
She highlighted a very, important dilemma we have. I will say the decision for an allo transplant is probably very controversial amongst lymphoma doctors. I think you have some people who are very beholden to it, and others who probably would never send a patient for an allo transplant based on what Robyn has already mentioned, the simple fact of, again, the risk of graft versus host disease and finding an appropriate donor. And then with lymphoma, actually making sure the cancer is in remission before you get to an allo is very important things. I will say, I wish we had a better overall system to illustrate clinical trials throughout the US. I don't think we still have a great system even in 2023. I think a lot of us depend on clinicaltrials.gov or word of mouth versus having a good system where any patient can just plug in their disease and, we pop up all these studies and trials that are actually active and open, because that's the thing about clinicaltrial.gov. Just because they list sites doesn't necessarily mean those sites are actually up and active, which I think a lot of people don't realize. So
I'll just agree with everything Tycel said, and also say that, Robyn was either, or maybe a combination, either real lucky with the choice that she made, or real prescient, or just real smart, because in retrospect, if it was me, a CAR T versus an allo transplant, it would be a no-brainer. And in fact, now we have randomized trials comparing CAR T to autologous transplants for those folks that relapse quickly, those first 12-month relapsers after frontline therapy. And now it's just about a fact that CAR T is probably both more effective and probably kinder and gentler with less of those toxicities. CAR T can be toxic, but overall, head-to-head, I would probably rather get a CAR T-cell therapy than an autologous transplant. I think most people would probably agree. And it's real tricky because Robyn hinted to that maybe some of the doctors recommended the transplant. Tycel and I and all regular doctors, we have a funny thing we say about surgeons. We say, when all you have is a hammer, everything looks like a nail, meaning that if you're a doctor of some specialist, you think that's the way to go. So a transplant doctor will say, yeah, we could do transplant here. And it's tricky. It's the same with clinical trials. If you're a doctor that doesn't, focus on clinical trials, you won't think of that. It won't come to mind. Robyn is one of these rare people that can advocate very well for herself. You do need doctors sometimes to be able to help advocate for you.
Love that, Dr. Brody. Thank you. Dr. Brody, let's talk about CAR T more in depth now, since we've already talked about it. But what exactly is CAR T cell therapy, and what does it entail in terms of how people actually go through it?
It sounds like science fiction, but literally we take a little bit of a person's immune system, some immune cells from their blood. In the old days meaning 3 years ago, we shipped them off to Santa Monica, California, and they literally do gene therapy on your immune cells. They put a new gene into your immune cells, and that gene allows when we put those immune cells back into you to go and first find lymphoma and then kill lymphoma. And humans have about 20,000 genes. This gene that they stick into those immune cells is not one of those 20,000. It's a made-up gene. We call it a Frankenstein gene. It's amalgamated from 5 other genes put together into one gene. That gene is called CAR, a chimeric antigen receptor. You get some of this flu-cy chemo, and then we reinfuse the CAR T cells. Very elegant science fiction, but it's here today. And just as a punchline of the efficacy, a few years ago, we would say folks with multiply relapsed DLBCL didn't have curative options, just like patients with metastatic breast cancer. And in those patients, we would say the CAR T cells might cure 35, 40% of patients. Going from what we call 0% to 40%, that sounds like it's miraculous. I mean, define miracle however you want, but it's a miracle if you don't think we can separate an ocean with our hands. Yeah, because it just can't be done. Well, you can't cure an incurable disease, except they did. They cured 40% of them, but still not 100%. So still a lot of room for improvement. The efficacy is therefore, I would say very impressive, miraculously impressive, but it's not benign therapy. There's this risk of side effects, which are real. And probably, maybe 20% of people get some of these high-grade side effects. And the most commonly discussed one, we have a fancy name called cytokine release syndrome, CRS. It's kind of like having a terrible infection, but there's actually no bacteria, no virus infecting you. And people can land in the intensive care unit because of that. And another one we just call neurotoxicity, and we sometimes call it ICANS, ICANS. We just call it neurotoxicity. And it can literally be a type of encephalopathy. People can literally start hallucinating, become unconscious. It can be dangerous. And again, these high-grade toxicities, one of those two, might happen in up to 1 in 5 patients. And even if it doesn't happen in the other 4 out of 5, we don't know which 1 in 5 is going to happen to. So we have to monitor all of those patients very closely, usually in the hospital.
Well, with that being said, Dr. Phillips, if you could just touch on the benefits and the challenges.
I mean, I think that is the biggest excitement, is that, again, we've been all talking about immunotherapy for decades. And here we are with a true immunotherapy with a much better safety and manageable safety profile than what we typically would see with an allogeneic stem cell transplantation. But I think to the point of the patient is what we see why the uptake of CAR T is not what it should be. I mean, I think the last time we looked, 25 to 30% of patients are probably getting CAR T that are eligible. That means the vast majority of patients are not getting CAR T due to access issues, meaning they can't get to a CAR T center, or they can't necessarily they don't have anybody that needs to be there with them or relocate to a center for a month to get CAR T. So, I mean, these are all logistical challenges that we are trying to sort of fix and overcome, along the way, but it's not been necessarily the easiest thing to get over, because these patients will need caregivers, and especially as we're trying to move CAR T to an outpatient setting. Again, I mean, they have to have somebody there that can take care of them, just because financially, I don't think CAR T is going to be feasible moving forward to stay where it was originally, where patients are hospitalized for 30 days, because fortunately payers aren't going to pay for that. And so hospitals can't fit the bills for that any longer. So they do need some sort of support to help take care of them, which is not, unfortunately, dissimilar to what we see with transplant patients. But I mean, the hope is that, that requirement and things like that may ease for some patients who aren't necessarily going through complications. But right now, unfortunately, it is a requirement at most centers that they have somebody to take care of them.
I have a general question about refractory patients, right? So different in that whatever treatment, there's no response. It's not like you were in remission and then you relapsed, it just didn't respond. So when would you consider CAR T for someone in that position?
Thankfully now, in second line, so that generally would be the immediate treatment at this point, unless there's some reason, that they can't get the CAR T. Those patients should be the patients we get to CAR T, because we already have 2 studies that have demonstrated that CAR T was superior to autologous stem cell transplantation with both axi-cell and then lyso-cell in this patient population. So this is the ideal patient population for CAR T. So easy answer, CAR T.
Now let's get into bispecific antibodies. Dr. Phillips, do you want to start off by sharing more about what are bispecifics?
Yeah. So the easiest way to think about bispecifics, I try to it is basically like a off-the-shelf CAR T product, in the sense that it still works to utilize your own T cells to fight off the cancer. But instead of the manufacturing process, it basically administers an antibody that will bind to CD20, but then also bind to CD3, or it can bind to CD19 too, with blinatumomab and some of the newer agents. But again, it'll bind to some marker in your cancer, then bind to your T cell, which will force an interaction between the two, at which point, will lead to T cell activation, and we hope some sort of T cell expansion, growth, etc. So in that way, shape, or form, these antibodies, at least the CD20, CD3s, and some of the newer CD19 CD3s, are a bit better than what we have with blinatumomab, which was given as a continuous infusion. By allowing those FC portions and just sort of muting them to be still intact to the antibodies, they have a much longer half-life. So these things can be administered more conveniently. So if we look at the agents that we have that will likely get approval, we have mosunetuzumab, which is approved for follicular lymphoma, which is given, as a step-up dosing once a week until you get to full dose. And then thereafter, once you reach full dose, is given every 3 weeks. For either 8 or 17 cycles. We have epcoritamab, which will likely get an approval for diffuse large B-cell lymphoma, again, given up in a step-up dosing fashion every 3 weeks. But that one continues weekly through the first 3 cycles, and bi-weekly from cycles 4 through 9, and then thereafter once a month indefinitely. And then glofitamab, which is another CD20, CD3 bispecific antibody, again, given a step-up dosing. This one is given with a pre-medication with obinutuzumab to help reduce some of the risk of CRS, which is still prevalent because it's a T-cell directed therapy, and then given once every 3 weeks thereafter. So this one is also given for a fixed dose of just 12 cycles. And then we have odronextamab, plamotamab, along in the pipeline of other sort of bispecific antibodies. And there are many more coming down the line. I think the key thing with the bispecifics, at least from what we can see from the early stages, is that they appear to be effective in patients with relapsed refractory diffuse large B-cell lymphoma. Some studies have shown that they are effective in patients post-CAR T. I guess the question that we'll get into later is, which post-CAR T patient is actually a patient that's going to respond to a bispecific. But still the same side effects with this sort of exist, that, there are CRS that occurs, probably not to the severity and intensity what we see with CAR T. And the management strategy will probably end up being a bit different. And also there is the ICANS and neurological toxicity, again, not to the degree and severity that we see with CAR T, but again, still present. So, I mean, a lot of, community physicians and patients will get firsthand experience, good or bad, with these bispecifics. And hopefully, more guidance and things will get out there for how to best manage some of these events, because a lot of these physicians haven't dealt with this. But I would say in a couple of years, hopefully, as more and more physicians get comfortable with these drugs, they'll get more access to patients and they'll be more comfortable managing the side effects. And this will probably sort of have a bigger catchment area just because of less restrictions on these drugs than what we see with really CAR T at this point.
Thank you, Dr. Phillips. That was a great summary of, again, a huge space. So appreciate that. Dr. Brody, could we talk a little bit more about, Dr. Phillips talked about the management overall. Could you give a picture of, so it sounds like for DLBCL in particular, there are two that are a little bit more advanced in the studies than the rest, although there are many that we should be watching out for, but epcoritamab and glofitamab are sort of the two that, again, are more advanced with the research. Anything in terms of the comparison between the two, what you see with the response so far with the clinical trials?
I will say overall that these two are much more similar than different. The easiest example of that is that they both have been in patients of, they've been in many hundreds of patients, but they focused on a trial of about 150 patients for each of them. And they both had complete remission rates of about 39%, 39.4% for both of them. So that's pretty similar. That means, again, just under 40% of people, not only does a tumor melt, but it disappears so that we cannot even see it on a PET scan or a CT scan. We don't want to confuse complete remission with cure. Just because we can't see any cancer doesn't mean there might not be a little bit left, but it does seem like a lot of these complete remissions are very durable, very long lasting. And we definitely have patients in complete remission from these therapies for more than 2 years. I mean, we don't have 5 years yet because these medicines haven't been around for 5 years really. But if a person stays in complete remission for 2 years, the chance of being cured seems to get to be pretty good. That's true for many therapies, certainly for CAR T and for other therapies. So they're more similar than different. One little difference between the two of them is the way that they are administered. Epcoritamab is given as a subcutaneous shot, just a shot under the skin, so pretty quick. Glofitamab is given as an infusion. The first infusion is 8 hours. Later on, it becomes 4 hours and 2 hours. So, a little bit more time intensive there, but not too bad. Maybe that's a point in favor of epcoritamab. I'll give a point in favor of glofitamab. To be fair, the glofitamab studies are mostly designed to be, we say, time limited. You get 12 cycles, about 9 months of glofitamab, and then you just stop the therapy. And if you're in complete remission, we just keep an eye on you. And if you were to relapse, you could get more of it, but if you're in complete remission, we just stop giving the therapy. That's just how they were designed. So we don't have to per se give it forever. The epcoritamab studies have been designed, just keep going with it. As long as the person's in remission and doing well, just keep giving it. Hard to say which one's better and worse. I find half of my patients like the idea of, give me a break. I've been through a lot. And half the patients say, I don't know, I kind of want to keep getting this therapy. We don't know which one is right or wrong. We don't know, but some people see the benefit of time-limited therapy, so they don't have to keep coming into the doctor as frequently. So maybe little benefits of one over the other in both directions, but overall more similar than different, than overall remarkable efficacy and much easier to use than CAR T cells. And really, I think both Robyn, maybe Joy, and one of the other questions was, if I do not have a caregiver and someone's recommending CAR T, what kind of options come up there? And the one-word answer is bispecifics.
Assuming bispecifics are, approved and it's not being used as a clinical trial for somebody. What about the side effects, Dr. Brody? Dr. Phillips did talk about CRS seems to be a little bit, lower grade or better, in bispecifics as opposed to CAR T. Anything else?
Yeah, as Tycel said, conceptually, it's the same possible side effects, but thankfully, we've mostly just seen much less of them in lower grade versions of them. One way to punchline this would be to say that I think the average CAR T cell patient today is still spending sometimes 11 days in the hospital just for observation, a lot of it, whereas the average bispecific antibody patient usually spends 1 day in the hospital. And again, even though the vast majority of them don't get any of those side effects, we have to, at the moment, still observe them all just to make sure that the 1 out of 10 that gets the bad side effect, we don't miss them. So the plan with most bispecifics still for DLBCL is at least one day of observation. And again, probably about 5% of people can get the high-grade version of that CRS, and probably much less than 5% get that neurotoxicity, we also call it icons. So rare, but it can happen, and therefore requires a bit of monitoring.
How do you consider which one to go to first for relapse refractory patients? And yeah, what are the considerations there?
Specific to this question, I mean, I think CAR T has the more definitive data. So I think if you have access to both, I think you're hard pressed to put a bispecific before CAR T at this current date. So I would say CAR T and then bispecific thereafter, pending the response to the patient with the CAR T. I would say if the patient has no response to CAR T, I have very little faith that they don't have a response to a bispecific. We feel much comfortable if a patient had some sort of response to CAR T and had lost it, to try to resurrect that response with the bispecific. So, but I think, like I said, if I have access to both CAR T, if I can't get CAR T, then ideally, I think a bispecific is a good sort of replacement in that situation.
I think it's actually less of a question of which is better, CAR T, bispecifics, because the numbers are that the majority of patients, frustratingly, for all the advances that we're bragging about, the majority of patients are not cured by either. So statistically, you would want to get both. You'd want to have the option to get both. And as Tycel just said, if you get CAR T today and they fail in 30 days from now, quite honestly, you probably won't be able to get bispecifics because there's no trials really that allow the rapid, failing CAR T patients to get access to bispecifics. But conversely, we've done the opposite in our patients quite a bit, where they'd got bispecifics. If they stayed in complete remission, they kept going, but we had a few where they progressed eventually, and we gave them CAR T cells.
Well, I like that we're comprehensive about this. I know we're almost at time. If you have a couple more minutes after, we have some audience questions, but before, since I know some of the participants and attendees have to go, Robyn, if each of you could we could talk about clinical trials really quickly, because I think there's a lot of misunderstanding. So as a sort of last point before we get to Q&A, Robyn, from the patient perspective, you went through a clinical trial. Anything you want to say about that in terms of, I think people think of like, oh my God, there's a placebo, or I'm going to be in an experiment, and there's a lot of fear. What would you like to tell people about your actual experience with the clinical trial?
Well, as a patient and also as a physician, I think it's important that people realize that clinical trials in a lot of cases are some of the best medical care that you're going to receive. They're the cutting edge, and it is often the way to go in unusual cancers that can't be controlled or by anything else, or actually some other diseases.
So I think that's the thing people need to remember. It's not guinea pigs. This is how we advance the field, especially in diseases without standard of care. I mean, this is really your chance to get something that potentially could be life-saving. So I think more people should be offered clinical trials and probably not, versus us misabusing clinical trials. That never is a scenario.
So what I want to tell people is keep your options open, keep your mind open, do some research, read. In this case, this is a blood cancer group. Talk to LLS if perhaps you've relapsed, see what their opinions are, and actually get several opinions. I do think you need to be your own advocate. We have physicians here. Physicians don't agree on things, and that doesn't make somebody wrong or right. It's a process. Science is not exact. It's not black and white. So you need to figure out what's right for you, do the research, and don't be afraid, because a clinical trial might be the solution for you.
Thank you. Robyn, thank you so much, Dr. Tycel Phillips, Dr. Joshua Brody, and Robyn, by the way, with the MD behind that, she's also a doctor. Yes. 3 rock stars. Thank you so much for making this such an enjoyable conversation about what could be very overwhelming.
Bye-bye.
Thank you.
Thank you so much.
Great job.